Hello everybody, my name is Jeremy Hecker and my project today is Situ microbial cultures of natural groundwaters using a serpentine substrate. The mineral serpentine is a metamorphosed product of olivine hydration. Olivine is a common mineral to Earth's mantle. Sometimes through tectonic forces, that olivine can be uplifted through Earth's crust and deposited onto the surface. An example of this is at the Coast Range Ophiolite at the McLaughlin Reserve in Northern California. The olivine has reacted with the groundwater in this location to create the mineral serpentine. The process of serpentinization involves gases such as hydrogen and methane. The groundwater in this area, due to the process of serpentinization, has become extremely basic, with an average pH of 12.5. This area of serpentinization is significant because orbiting image spectrometers have found areas such as this on the surface of Mars. The objective of my research is to understand the relationship between this environment and chemosynthetic microbes that may inhabit these environments and utilize the products of the process of serpentinization for energy. For this research, I have chosen 7 types of water and 6 atmospheres that are experimentally relevant to the process of serpentinization. The atmospheres we chose was a vacuum, hydrogen gas, carbon dioxide, hydrogen and carbon dioxide, methane, and hydrogen and methane. Each of those atmospheres was inoculated with a different water. DI water as a control, surface water from the McLaughlin Reserve, mainly rainwater, and 5 different CROMO wells. After incubation of approximately 30 days, testing began. DAPI counts were attempted to determine growth yields, however the lack of concentration in the samples made this unreliable. Instead, ultraviolet visible spectroscopy was used to determine nucleic acid concentrations as a method of determining growth yields. A scanning electron microscope was also used to try to determine the relationship between microbial chemosynthesis and minerals. The graph you see behind me is the results from the UV-vis spectroscopy, which show growth yields for most of the environments. A high yield in the methane environment with similar yields in the vacuum and hydrogen and methane environments, and some yield in the carbon dioxide environments. Low growth was observed in the hydrogen environment, and no growth was observed in the hydrogen and carbon dioxide environment. The SEM images here may show microbial adhesion to specific minerals. We think they might be utilizing iron-rich minerals for metabolism. Further research will be used to determine if that is the case. The 3 images below that are images using DAPI stain and may show microbial communities or microbes encapsulating clay minerals for metabolism. Now that microbial growth has been determined in our samples through the use of UV-vis spectroscopy, further research will assess whether or not those microbes that exist in those different environments are the same or not, or whether the growth yields are due to the fitness of one particular microbe. Terminal restriction length polymorphism is a technique used to lyse DNA at particular base pairs into different lengths, creating a rudimentary fingerprint of the DNA of the microbes. If they're found to be the same in the samples, we can say that one particular microbe has an affinity for a particular environment, or that different microbes are populating in different types of environments. And additionally, samples of the remaining wells will be analyzed for similar results to see if the aqueous chemistries within the different wells has an effect on microbial growth. So thank you for listening to my presentation on H2 microbial cultures.